So hello, I've introduced myself a few times, so I will just jump right in and say I'm excited to be talking to you all today about Puget Sound zooplankton monitoring. This is a big collaborative effort. You can see by the list of people that I work with within the Nisqually tribal staff, including Nano Perez, Eddie Villegas, Kyle Kautz, Sam Stepeton, Walker Duvall, Renee Bracero, and Tom Frederick. Those are all people, and including Ken Moore too. I think he's been out for some zooplankton sampling. Those are the people that have been out on the boat with me in over the last couple years. And then there are the people who do what I consider to be the really hard work, the counting and identification of zooplankton. Those are University of Washington research and laboratory staff. Julie Keister is the primary investigator in this program. She's a PhD at the University of Washington, a professor. Amanda Winans, Bethany, and Olga are the other techs, and I think they're probably also training up students every year. They are probably training up a new batch of students to help out with this work. And then the field work, in addition to the Nisqually Indian tribe, you can see there are a lot of other logos and tribal governments and state agencies, and even King County that are all involved in helping to do this work. So it's a big effort. There'll be a map eventually, and you'll be able to see the scope of this work across Puget Sound. A little bit of an introductory of myself. I really enjoy both professionally and personally the Puget Sound. I'm lucky I get to spend a lot of time on it, and walking along beaches, there's always a lot to see. On a boat, there's a lot to see, and I'm hopefully this maybe work will encourage you to think even about the small stuff that we don't see so easily. Professional interests, a few others: marine and freshwater habitat restoration. Habitat restoration has been increasing in popularity and prevalence over the last 10 years, probably a little longer than that. There's been increased funding to it, and I think that's a really important and promising development. I also enjoy tinkering around with new technologies and new monitoring efforts. Like zooplankton monitoring, this was a new project. A lot of people were kind of engaging in it for the first time, and I enjoy scenarios where it's accepted that there's going to be some trial and error. Having that space to operate and learn is definitely, I enjoy it. And another project I'm working on is using imaging sonar to count salmon in highly turbid rivers like the Nisqually that melts off of a glacier and brings down a lot of really glacial flour that makes the river really difficult to see into. Personal interests: I love spending time with my family, my parents, in-laws, Megan, and our two kids, Cormac and Riley. And sports, and I heard somebody else say gardening is really fun. I enjoy it too, but I think my favorite part of the gardening is the food element. So, starting at the beginning, plankton can be defined as a diverse collection of organisms found in the water that are unable to propel themselves against a current. So, a lot of these animals, these zooplankton in this picture, you can see can swim, but relative to their size and the speed of the currents in which they exist and live their lives, they don't propel themselves very well against that current. So, not to too much discredit their swimming ability, but if you wanted to describe zooplankton or plankton in general, you would describe them as mostly small and weak swimmers. And when I say small, inside of that red little oval at the bottom of this graphic, that is a 100 micron line for reference. So up in the top left corner of this picture, you can see that purple and orange copepod with its big trademark antennae swimming out, which they actually use to swim as well as detect and sense the environment around them. That is about 100 microns wide. And the image in the bottom right here, that is a larval crab. That's the megalops life stage. A coworker of mine kind of referred to all of these zooplankton as dinosaurs. And if you imagined these things as big as we are, they would probably be pretty intimidating, pretty startling. So zooplankton are a type of plankton. They are the animal plankton. Zoon is Greek for animal, and planktos. I actually didn't know this until I did a little research for the presentation. Planktos means wanderer or drifter. So that's kind of a fun one. And phyton of phytoplankton is the plant plankton. And on those images on the right, you can see lots of chlorophyll. Several of those types of phytoplankton are diatoms, which live in a glass shell. That's kind of their form of a supportive structure that allows light to come in. So yeah, clever little plankton doing really cool stuff. So why do we sample zooplankton? Zooplankton, for quite a few reasons, are just right. They are both a critical food source in the food pyramid, so they're relatively low down on the pyramid, but they're not so low in this food pyramid that they are tiny and really difficult to count. Phytoplankton, some types of phytoplankton are very small. We saw in some of those previous pictures some diatoms. Those are one of the larger types of phytoplankton. And because zooplankton depend directly on phytoplankton as a food source, their abundance, the abundance of zooplankton, rises and falls with the abundance of phytoplankton. There's a little bit of a lag, but they're a really good indicator of how much productivity is going on photosynthetically. And luckily enough, in addition to these other elements, zooplankton are eaten by baby salmon, which makes them important for salmon recovery biologists like me. Another thing that is eaten by salmon is herring. So herring at their different life stages are eaten by different sizes of especially Chinook. But because marine food webs are funky and a little bit surprising, I posed a couple of questions here. And the first one is kind of intuitive. What type of zooplankton do herring eat? Well, the big ones, of course. Like we all have that instinct of if there's 2 apples or 2 of something and we're feeling hungry, we take the bigger one. The less intuitive is probably what type of herring do zooplankton eat? And what are zooplankton doing eating herring anyway? Well, it turns out that some of these different types of worms that are small enough to be eaten by the larger sizes of herring are able to eat herring eggs and herring larvae at their smaller stages. So the zooplankton, it's not all one-sided. The zooplankton do get their turn in the food web, which brings up the fact that marine ecosystems are complicated. In the simpler diagram on the left, you see we have about 16 species here, and the generalized, simplified predator-prey, arrows pointing to the predator and away from the prey in the interaction, we have 28. So that's kind of manageable. We can kind of wrap our heads around that when we look at it. And this diagram helps us understand how PCBs move up the food chain, pollution. But when we look over at the diagram on the right, we see 50 plus species with quite a bit more than 100 links. This type of model takes a computer to adjust for increases or decreases in different groups of these species. So there is a lot going on out there. It's one of those classic life and death scenarios in nature. Everything's eating everything else and trying to avoid being eaten while it's doing it. So all zooplankton do not stay small. You can see a larval sand dab on the left, almost completely translucent. Just the little eye spots and some of the nerve bundles around its lateral line, and maybe a tiny bit of pigment associated with the fins help it stand out under a microscope. And then we see the diverse and kind of bizarre shapes of juvenile crab. The first one is a zoea, the first one on the top left of that crab set of pictures. And then the one to the right of it, the one in the middle, and the one with multiple little crab larvae, that is the megalops stage that we saw on the first introductory picture. And eventually they grow up to be a crab that we would recognize. This is a Dungeness. We also, while we do catch some Dungeness larvae, we also catch shore crab, graceful crab, some hermit crab. They're actually I shouldn't even guess. I'm not a zooplankton expert, but there are many, many types of crab in the Puget Sound. I think it's probably over 24, I would imagine. So how do we sample zooplankton? We use two types of net. We conduct oblique tows, which are done diagonally through the water. We go forward and drop the net down to 30 meters, and while we're going forward continuously, we then raise it back up. That is the black net on the left. It has a larger mesh size, so it can filter more water, and it is limited, though, to only catch some of the larger species. The vertical net, we come to a full stop and drop it straight down in the water and try and get it pretty close to the bottom, usually with a 10 or 15 foot buffer margin of error. And then we draw it straight up at a nice even rate. And we sample the entire column of water from close to the bottom of the water, close to the bed of the sound, all the way to the surface. This one has a smaller mesh size, 200 microns, and catches the smaller species and even some of the larger diatom phytoplankton species that we saw earlier. We sample, most locations are sampled once every 2 weeks from March to October. The Nisqually tribe and King County zooplankton samplers are out there throughout the entire year, because there are some interesting things going on in November, December, January, February. Different species are present in the plankton during those times. And here is a higher resolution picture that's easier to see the components of these nets. The flow meters, there's one in each net. Those measure the amount of water that has passed through the net, and those are really cool. And I'll tell you why in a little bit, but just stay ready to hear about how awesome flow meters are. The frame holds the soft and flexible net open at a uniform size. The mesh is kind of the workhorse. It's what holds the animals in and lets the water through. And the cod end sits down at the bottom, and that is where all the sample gets rinsed down into. And then the extra water can be filtered out, and then the sample is poured into a jar and preserved. So these are some of the similar groups from above, from our first slide, showing who conducts the sampling and where. Each of these yellow dots represents a location where the bongo net and vertical net samples are both taken. Some of you are probably noticing the red dot. At that location, bongo net and vertical net samples are taken, and some other oceanography is conducted there to measure nutrients in the water and water temperature. And then again, we have the sample analyzers who, if it wasn't for them, we really wouldn't know very much. So thank you, School of Oceanography. So what is it then that we are learning from all of this sampling? We learn species diversity, or the species that are present. So what types did we catch? What types were in the water at that location? We learn species abundance. How many of each type of zooplankton did we catch? And you can start to see why the sample analyzer's job is so time consuming. And then also from that, we measure weights, and we get species, they measure species biomass and total biomass. This gets us a general idea of the calories and the types of nutrients that are available to contribute to the food web in those locations at those times of year. And this is why flow meters are so cool. We know all of these features per gallon of water that we are sampling. You'll see in some of these other charts that, well, let's go to the charts. If you look at the y-axis, you can see that the abundance, these counts, are relative to 1 cubic meter of water. I think that's really cool, because if we have an estimate of the volume of water in Puget Sound, it's a pretty simple multiplication exercise with lots and lots of zeros to describe how many zooplankton are in Puget Sound. And you can see if 1 cubic meter has 10,000 zooplankton in it, that is an amazing amount of photosynthesis and predation, and base of the food web is going on, intensively all around us. Really cool. You can see also some patterns from this data. The x-axis across the bottom is either July or January, so summer or winter of the years starting in 2014, when we started doing this work, through 2020. So some patterns you can see is that there is quite a bit higher biomass and abundance in the summer than there is in the winter. A big reason for this is the availability of light and longer days in the summer, and quite a few of the species of plankton that we are sampling do have some low temperature limitations. So when it's cold, they don't divide or grow nearly as fast as when it warms up a little bit. We might not say that 45 or 50 degrees Fahrenheit water is warm, but to the zooplankton in Puget Sound it is, and they really get fired up, even in even in March when it warms up just a little bit, they get rolling. So if you prefer circles, here's a completely different way to think about average monthly biomass. Um, you can see along the top row, each column running top to bottom represents a month. And each row along the side, you can see represents a year. So if we start in 2014 on the left side and look over at May, we see that a lot of those circles are about the same size. That means the average monthly biomass of zooplankton in the vertical toes was relatively similar at all the sites in May in 2014. Go down one square from there, though, we see in 2015 the northern Puget Sound, just a little east of like Port Townsend there's a really big circle. That area experienced some real productivity in May. So then if we stay in 2015 and look to the right at the orange circles, we can see that average biomass came back down by June. So we see from this that sometimes you can have a real boom of productivity, but that doesn't guarantee that it's going to last. And there are lots of interesting patterns if you look at this slide for a while and spend some time with it. There's lots of interesting patterns here, and I encourage you all to do that if you want to download this presentation and spend some time looking at all this work. And now that you're familiar with how to look at this type of data, here are some pie charts of crab species. And again, you can spend some time with this and see what it all means. Maybe look over at the Latin genus and species names on the right and figure out if one of your favorite species of crabs is over there, and see what season and what region they find the greatest abundance. One thing I should reference too is that gray blob underneath all these circles is actually a map of Puget Sound. So similar to the orange dots showing where the different groups conduct their sampling, that is a map of where that sampling takes place. So you can see if you go to June 2020, that blue, mostly blue and a little bit yellow circle, that is South Puget Sound and the Nisqually zooplankton sampling area. So if we were going to figure out which species of crab, maybe you can do that when you spend a little bit of time looking at this in the future. All right. So what can zooplankton tell us about salmon and their survival? Well, based on similar work that was done in the Pacific, we are able to compare zooplankton species abundance and trends to the survival of coho and Chinook salmon, which we're measuring by other means, whether it's measuring their growth or measuring the return of coated wire tags to hatcheries from year to year. But this graph right here in this line that fits the data at a relatively good, it's a relatively good fit. This graph shows us the relationship between zooplankton abundance and other traits and coho survival. So we know that when the ecosystem indicator is strongly negative to the left of this graph, that coho survival goes up. So this helps salmon harvest managers and recovery biologists like myself better predict the strength of a salmon return in a specific year. And one thing to notice is how many dots there are. I counted them, and there's 15, except if you see down in the notes here, 2011 is excluded from this data set. So there are 14. So it took 14 years of sampling, including one anomalous year that they felt like didn't really contain very useful information, to get this trend line that they felt comfortable had isolated the signal, which was the trend line, from the noise of the patchiness. If you look at -2 along the x-axis, you can see at some values of -2, some years, survival was not as good as others. Some years you can see survival was at 3, and then there was one year it was at 4. So you need more than 1 or 2 or 3 or maybe even more than 10 samples to really get confident in that relationship. So can we identify similar patterns in Puget Sound? Yes, we can. There are some interesting potential patterns that we think are emerging. And then sometimes when you think you see something, a year of data pops up that defies that trend. So then you have to wait for a few more years to see if that trend holds going forward. But every year of data does get us closer. And if you enjoyed this presentation, then you would probably want to check out the Puget Sound Partnership's YouTube channel. And this specific address is to a really nice video that they did that looks more in depth at the zooplankton sampling project and some of the other project partners in addition to the Nisqually tribe. And maybe that's a link then Tristan might want to pull out and make more clickable and easier to access. But yeah, there you go. And I'd be happy to answer any questions that people have, or maybe I have said enough words and you're all comfortable with it. But if you have any questions, I'd be happy to talk more.
Well, thank you for such a great presentation. A question I had was, is there can we tell the trends for future zooplankton in kind of, I guess, the Salish Sea and the Puget Sound, or is it kind of just something that fluctuates year to year?
That is a great question. We are seeing some trends in zooplankton productivity and phytoplankton productivity related to water temperature. Some of those are relatively well known by species specifically. Some of the trends that we're seeing, people were not expecting, though, and those are trends that operate among the system as a whole. It's not just one species responding, it's some feedback loops, it's some amplification of trends within the data. So one thing that would help and that we are interested in expanding is our measurements of water temperature and salinity and nutrients and some of the energy and mixing of the water in the system. And as we add that to our data set, we will then be able to say if it's an El Nino or a La Nina, or there's a marine heat wave, or we're going to have a really hot summer and have winds, blowing from inland offshore, we can start to predict what that might mean for zooplankton, and then what that might mean then for salmon also, if we want to take it that far. But yes, there are predictors of zooplankton abundance and health, and the varieties of zooplankton and what their relative abundance is likely to be. Is it going to be, are we going to have more warm water zooplankton or cold water zooplankton? Yeah, thank you.
I had a question about how climate change is affecting your research, if it is affecting it.
Yeah. There are likely trends or influences of climate change that are happening right in front of us. And as this is a relatively new, kind of young data set, we may notice that some of the early years have a different characteristic than the later years, subsequent years. One likely trend of climate change that we saw was the marine heat waves of several years ago. Those did make Puget Sound waters warmer. That did initiate some phytoplankton and then subsequently zooplankton abundance peaks earlier in the year, warmer earlier, growth earlier type of scenarios. Exactly how those fit into some of the more predictive patterns, we're not quite there yet, but that is likely something that the School of Oceanography and those sophisticated researchers will tease out in the future. And one, warming is something that resonates with everyone. We all pay attention to water temperature or air temperature in our day-to-day life. The other thing that is going on in marine waters, especially that a lot of these animals are sensitive to, is ocean acidification and the pH of the water and their ability to grow and form shells of different substances in the presence of a lowering pH. So some of those things are probably just happening right in front of us, and we don't have a long-term data set to exactly pin them down and describe how things are changing relative to how they were before. But thank you. That's a great question too.
Yeah, thank you.
I had a question relating to Dominique's. You mentioned that the zooplankton become more abundant in warmer water. I've also learned that sometimes colder waters are more productive due to oxygen richness. How does that balance out in different species? I guess some like it hot and some don't, huh?
Yeah, I would say it's more relative to nutrient density. Oxygen is another story, but often the colder water, especially offshore in the ocean, is brought up from deep below the ocean surface. So it's really deep, cold, and kind of older water that has been down low for a while and has been nutrients have been absorbed by that deep water from the sediments down low as they're decomposing. So when that cold, deep water comes to the surface, it brings with it a lot of nutrients, the nitrogen, phosphorus, sulfur, iron that the phytoplankton really are craving and will respond rapidly to. Mixing in Puget Sound kind of results in there's a decent amount of nutrient abundance in Puget Sound. Also, sewage outfalls put a lot of nutrients into the waters of Puget Sound. So nutrient scarcity is not quite the driver of boom and bust like it is in the offshore ocean. Another thing too, though, that is actually in a different slide that I didn't show, but it's a pattern that has was discovered by this other work in the Pacific Ocean. Cold water copepods are both bigger and have a different lipid content than the warm water smaller copepods. So cold water copepods are more nutritious and require less energy to forage on for these fish. You get more nutrients per bite when you're eating those when salmon are eating those larger copepods. So just because the productivity ramps up, it can kind of be iceberg lettuce versus, romaine or cabbage or something more substantial. It's not always more is better. Sometimes what's in it matters too.
That was an amazing answer. Of course, I'm biased.
Thank you.
All right, are there any other questions? All right, well, thank you so much, Dad. And we will hand it over to Megan. Oh, I think you're on mute. Yeah.
Oh, I was just talking about how Jed is a fisheries biologist and an oceanographer. Okay, so are you guys seeing, oops. Are you guys seeing the presentation or the slide sorter?
We're seeing your notes as well.
Okay. How's that?
There it is, yeah.
Okay. Okay, so I'm going to switch gears. Jed gave a really good introduction to what I'm going to talk about, although I'm going to talk about the top of the food chain rather than the bottom. So I'm mostly going to talk about harbor seal consumption of steelhead smolts in the Nisqually estuary today. I'm Megan Moore. I introduced myself. I'm a research biologist with the Northwest Fisheries Science Center. And these guys over here on the left-hand corner are the co-authors of the project. And I'm going to give a lot of the credit for this analysis that I'm going to talk about today to Austin, because he was the one who, during a Salish Sea Marine Survival Project meeting, realized that we had all the data already in hand to be able to make these calculations that I'm going to talk about today. And then these other guys, also Mike Malik, Matt Kungle, and Barry Jekian all contributed data and expertise as well. So Jed talked about the food web in the Salish Sea, and I also have an awesome figure from the Department of Ecology showing the species interactions in the Salish Sea. I'm going to talk about this particular interaction of harbor seal as predators of steelhead prey, and talk about how the abundance of top predators can really influence the prey that they eat within a food web. There's a really classic and well-studied example of the effects of top predators on the lower parts of the food chain. Sea otters are really well studied, and it has been shown that when sea otters decrease their prey, which are sea urchins primarily, they increase, and then the grazing of these sea urchins decreases kelp density, and then decreases the habitat for forage fish. So really, there's these really strong effects of these top predators. There's predators that are even higher in trophic level that affect sea otters, like humans through hunting, and killer whales that are actually limiting sea otter recovery in Alaska. So really, large predators can strongly affect their prey species. I'm sure many of you have heard that there's many large predator species that are in decline or struggling to recover. And that's still the dominant trend across the globe. But there are some notable exceptions, especially as there are some exceptions where species have started to recover from low exploitation levels. And this particular figure is from a Kamen et al. Paper showing that a number of pinniped species, which are both sea lions and seals, are actually increasing in abundance on both coasts of the U.S. And this line that's in each of these figures is showing that in 1972, the Marine Mammal Protection Act was enacted, which really enabled these populations to rebound. And in a lot of cases, they have rebound to carrying capacity. And the paper goes on to talk about some of the conflicts that have really started to emerge between these abundant predators and their prey that are depleted and struggling to recover. So one conflict that's emerging in Puget Sound is the conflict between protected harbor seals and listed Puget Sound steelhead. This figure is showing the decrease in abundance that we've seen in Puget Sound steelhead populations over the past 2 to 3 decades. Puget Sound steelhead have declined about an average of 50%, and the Nisqually River steelhead population has actually declined to the most dramatic extent compared to these other populations shown here. One of the primary drivers of the decline in Puget Sound steelhead populations is this decrease that we've seen in marine survival. So the survival of the smolts as they exit the river and go out to the Pacific Ocean and then come back to spawn. And you can see that it's gotten quite low in recent years. That trend is in direct opposition to the trend that we see in harbor seals, where you see increase in harbor seals. So there's kind of a negative relationship here between the predator and the prey. There's also a growing body of evidence of harbor seal predation on steelhead in the Salish Sea, and this slide is just to say that there's many other studies show that harbor seals eat steelhead, and it's actually listed as a primary pressure limiting the recovery of Puget Sound steelhead in this NOAA Fisheries recovery plan for Puget Sound steelhead. And we know, that this interaction is occurring. We just don't know what the magnitude of that effect of predation is on these depleted steelhead populations. So that was the question that we really wanted to answer with this analysis that I'm going to talk about. What percentage of the Nisqually steelhead run is being consumed by harbor seals in South Puget Sound? So that's what we set out to answer. And luckily we had a number of different data sources that we could kind of feed into these analyses. And to be really thorough, we used 2 different methodologies. First of all, this is a picture of harbor seal poop. And so this is the prey consumption model where we used harbor seal scat to understand what percentage of the harbor seal diet was comprised of steelhead. And then the second approach we used was a telemetry-based analysis on the behavior of the fish using these acoustic tags to monitor behavior and measure mortality in that way. So the 3 main data inputs for, first I'll talk about the consumption model first, and then I'll talk about the acoustic telemetry the acoustic telemetry method, and then I'll compare the 2 at the end. But for the consumption model, the 3 primary data inputs were the number of smolts that were entering the estuary. You have to know how many smolts you have, the number of steelhead eaten by 1 seal per month, and then multiply that by the number of seals in South Puget Sound. So you need an abundance estimate as well for that area. And the 3 primary outputs of this model were, and I'll talk about that in the results, we found the number of steelhead consumed, the percent of the run consumed, and the percent of the total South Puget Sound mortality that we can attribute to seals. So to find the number of smolts that were entering the environment, we used Washington Department of Fish and Wildlife operates a smolt trap, and they have an estimate of abundance at that location here down in the bottom of the slide on the map. It says smolt trap, and that's at river kilometer 19. And then to account for the mortality that occurs in between the smolt trap and the estuary, we used an estimate based on acoustic telemetry. We every year release steelhead smolts at the smolt trap and track their migration and measure their survival to the estuary, and then also from the estuary to this line of acoustic receivers that we have at the Narrows Bridge. So then we have the freshwater mortality, or the freshwater survival, and the survival from river mouth to the Narrows to account for the total mortality that's occurring as they migrate through South Puget Sound. So the second part of the model was to look at the, we wanted to calculate the number of steelhead eaten per month. And so that was a bit trickier. We actually collected, and Jed was one of the main collectors of the poop at all these harbor seal haulouts throughout South Puget Sound. They gathered up the seal poop and then actually used this technique called metabarcoding, where they extracted the DNA from the poop from all the different species that were eaten by the seal, and then were able to identify the fraction that was composed of steelhead. And that's what I'm showing here, is the percent of the diet that's composed of steelhead in each month of each year that we had samples. And so you can see that it's a really low percentage. But then we multiplied that by the energetic requirement of seals, which is actually 2.1 kilograms per day, which is quite a bit of food, and then multiplied that by the number of days in the month. And then we took that biomass and converted it to the number of steelhead eaten per month by dividing it by the weight, the average weight of those steelhead smolts that we tagged. So the third piece of data that we needed was a seal abundance estimate. And this we got from a paper that was published in just last year, and they took aerial counts of seals in Puget Sound. In South Puget Sound, we needed just the seals from that region to look at because that overlapped with the samples that we had taken. And that paper gave us the point estimate for each year, along with confidence intervals, so that we were able to look at a range of different abundances, the low, the mean, and the high for each year. So we fed, this is kind of like a schematic of the model, and kind of shows how we incorporated error into the model. We picked a scenario of seal abundance, and then we drew a smolt weight from the smolt distribution, and we drew a percent of the smolts eaten from normal distribution. And then we simulated that calculation 1,000 times to generate the range of different outcomes for that calculation. And then we summarized those by year for each seal abundance level. So here are the results from the model. So this is showing the number of steelhead that were consumed by South Puget Sound seals. And I'll just kind of talk about the mean for the mean seal abundance, the mean model outcome for the mean seal abundance level. So in 2016, there was about 15,800 steelhead eaten, 7,700 in 2017, and 40,200 in 2018. That doesn't mean a lot unless you look at it as a percent of the run. So we did that as well. So in 2016, there was 19% for the mean, 9%, so a lower percentage in 2017, but 33% of the run in 2018. So that means that 1/3 of the steelhead that are entering Puget Sound get eaten before they leave South Puget Sound. So we also were able to calculate the percent of the steelhead mortality that we can kind of blame on seals. And there's a lot of error. You can see these individual dots are the different outcomes, and they really range the entire length, the entire range of the percentages. It's pretty wide confidence intervals. That's because we had such low percentage of steelhead in the diet. But the mean value for this calculation was 67%. 26% in 2017 and 70% in 2018. So there's also quite a few outcomes where the model says that 100% of the mortality is attributed to seals. So that's not as easy to pinpoint as some of the other calculations, but it shows the that it's most likely a pretty good percentage in at least in 2016 and 2018. Okay, so now I'm going to talk about the second methodology that we used to measure mortality of steelhead due to harbor seals. And for this, we did, starting in 2014, we tagged steelhead smolts and released them in the Nisqually River. Each year we had between 98 and 204 in 2018. We had quite a good sample size. In 2019, we also used these V7T tags, which means that those tags had temperature sensors. And for each year, we were able to monitor their behavior as they migrated out of the Nisqually River past these receivers pictured here, these 3 pairs of receivers that we deployed in the same place every year. So we could really compare behavior among years. And we started seeing this really interesting behavior that didn't look like a steelhead behavior. And so I'll play this animation. Each color is a different tag. And this red tag is showing this tag going back and forth, not going down the river into the South Sound like it's supposed to, but going back and forth up and down the river. We think that these are tags that are getting eaten by a harbor seal, and the tag is in the belly of the seal. So the blue is going to show up. There it goes right out, and the blue and the yellow and the dark green are showing what it looks like for a typical steelhead to go out and migrate down the river. They just go straight down the river without going back and forth at all. These back and forth tags were never detected at any of the other receivers farther along the migration route. So we're pretty sure that these are mortalities. And so we can use that behavior to look at to really quantify how many of these steelhead are getting eaten by harbor seals. This is what it looks like in graphic form. So the survivors, this is time on the x-axis and distance on the y-axis. So you can look at the progression of these smolts, and it's very linear for the survivors. And you can see this very dramatic back and forth behavior for the mortalities. So we can calculate a percentage, and that's our mortality percentage that we can compare to the other mortality percentages that we have from the consumption model. But we also have another source of data in the temperature transmitters that allowed us to expand those calculations. So 91% of those smolts that were tagged with temperature sensors reached the estuary in 2019, and we got our normal back and forth count. But then we also saw that we could use the temperature tags as well to define those mortalities. So the survivors are showing this really consistent behavior, this consistent temperature reading of ambient river temperature, where they're just migrating down the river. But these mortalities are showing an increase in temperature as when they get eaten, and the temperature of the predator belly is much warmer than the ambient temperature. So we can use the temperature to discern between mortalities as well. And since we know the proportion of temperature tags to back and forth tags, we can account for those tags that were eaten but did not get detected as being a back and forth behavior in previous years. So we can expand the calculations that we made in previous years to include likely mortalities that we weren't able to account for. So these are our final numbers, and we can compare those to the model percentages. And these are so this is basically like the culmination of the talk, showing how these different mortality estimates kind of stack up against each other. So the blue is showing the back and forth, percentage. And the green is showing what the consumption model estimated for the mean seal abundance. And then this South Puget Sound is acoustic telemetry derived estimate of the entire mortality rate, not necessarily due to seals, just due to all the mortality. So you can see that the estimates of mortality based on the behavioral analysis are pretty much within the confidence intervals of the consumption model. So I mean, there's pretty good agreement between the two methods. We saw that the highest consumption mortality was in 2018. Perhaps that was due to the fact that we actually sampled more scats in the estuary, and we do see quite a bit of mortality occurring in the estuary itself. We saw similar interannual trends for both methods with this low rate of mortality in 2017. And then we saw different proportions of the mortality that was attributed to seals. So you can kind of look at this total mortality rate and then look at how much of that was described by the harbor seal mortality. And you see a high percentage in 2016, kind of a low percentage in 2017, and about half that mortality was accounted for in 2018. So we'll just kind of wrap up here with some general conclusions. We found through this analysis that harbor seals consumed between 9% and 33% of steelhead migrating through South Puget Sound. And assuming that mortality is additive and not compensatory, meaning that steelhead aren't going to get eaten by something else as they migrate through Puget Sound, we found that release from harbor seal predation would increase survival through Puget Sound dramatically, with these higher numbers of smolts making it to the marine environment. And despite these really low percentages that we saw in the harbor seal diet, the large number of harbor seals in the population in South Puget Sound kind of imposed heavy impacts on the small steelhead population that is coming out of the Nisqually. And we can't achieve a total absence of harbor seal predation, but I'm hoping that these model estimates can help managers kind of understand the severity of the issue that we're dealing with these high numbers of harbor seals and really low numbers of steelhead, and the impact that these large numbers of steelhead can have on these listed species, and hope to gauge whether predator intervention should occur in order to recover threatened steelhead. And that's kind of the perspective that I'm coming from. So if there's time, I will, oh, and I definitely want to acknowledge all of the help that that we had for all of these data that went into all these analyses, especially the Nisqually tribe, DFW, University of British Columbia, NOAA, Long Live the Kings, and the Salish Sea Marine Survival Project technical team. So thank you so much for listening, and please ask questions if you have them.
Well, thank you so much, Megan. That was incredible. That was an incredible presentation. I guess I'll start off with a question I had was, or we're talking about kind of the large predators declining. Is that what's playing into kind of this abundance of seals and sea lions, or is that more, I guess, yeah, we're seeing these like huge increases of them just kind of around the rivers and eating a bunch of steelhead. Is it that the predators can't find them because of boating traffic, or is it just they're declining and that's what's giving rise to these seals?
Yeah, that's a good question. So yeah, harbor seal populations were really low prior to 1972 when, because they had been heavily exploited by humans, and that's kind of the main reason why large predators are declining in particular, because of human fishing and hunting. But yeah, so after they were protected, they began to recover, and basically, you know, there's no management of them other than they're protected, and so they've kind of been able to recover. And it is a success story because, you know, a lot of marine mammals haven't been able to recover, like sea otters, for instance, haven't really been able to get a foothold in Puget Sound, but definitely the pinnipeds have. And that's true in Puget Sound and along the coast and in the Columbia. And so, yeah, it's kind of a regional, interesting problem to have when you have these recovered populations that are causing conflict. So it's difficult.
Yeah.
Megan, I have a question. I would think that there's a lot of different pressures on salmon that have waxed and waned, maybe mostly waxed, but I'm curious why before the pinnipeds had staged their comeback, that the salmon weren't doing better. And because I'm thinking a lot of the dams were already up, that had caused a major decline. What has happened, I guess, since 1970 to overall populations? I guess I don't know the answer to that question. Is it different now than then? What are maybe some of the other pressures? It's a really broad question, I imagine. You know, all kinds of barriers have been erected to migration that you know a lot more about. So maybe you can tie that all together with a bow.
No, I think that there are, I mean, my area of expertise is kind of looking at the pressures that are occurring in the marine environment. I started studying steelhead when we didn't know anything about what was going on in the marine environment. And now we do know a lot about how the survival through the marine environment is a really important stage in the life history, and there's a lot of mortality that occurs there, especially right when they enter. And then I think that's kind of normal, but the amount of mortality that they're experiencing due to predation is, I think, above and beyond what a normal ecosystem and a normal population should have to withstand if you're going to be stable, if that population is going to be stable. There definitely are still a lot of different pressures that continue to be threats for steelhead and pressures. But since they were listed, in 2007, there isn't a recreational harvest for Puget Sound steelhead. There are some harvest. I know that they don't harvest them in the Nisqually, I don't think. Some tribes do on a pretty low level. And in Hood Canal, I know there's not even any hatchery influence other than some kind of conservation hatcheries. So they've kind of tried to lift a lot of those pressures, but the recovery has not happened, in the last 15 years that they've been listed. So that's kind of one reason why I think the marine survival is really an important issue, because it's something that we do kind of semi have control over if we're going to manage this predator-prey interaction. Also, there's kind of mortality issues at the Hood Canal Bridge, which is something that's anthropogenically caused and also is within our control. So there's things that I think we can do, but I think it's very telling that we've kind of tried to lift some of these pressures on steelhead in particular, and that they haven't been able to recover, but rather they've actually gone down since the listing. So.
Thank you.
All right. Any other questions for Megan? So Jed also said there's no current hatchery steelhead influence in the Nisqually watershed as well.
Right.
All right. Well, thank you so much, Megan, for that presentation.
Yeah, thank you so much.
Yeah, of course. Yeah, it's very insightful.